Hey, welcome back to Bio 100. Hopefully, you guys are healthy and happy and everything's going well for you this fall in your other classes and in this one as well. So, today, I just want to jump right into it. We're going to be talking about Hardy-Weinberg theorem today and how to calculate it. Now, some of you I know immediately who've had are apprehensive, especially those who've had Bio 100 before, right? It's a theorem, there's clearly some math that's going to be involved, and you're concerned. But don't be. Just hang back. We've done this with thousands of students. We've broken it down pretty well. We're not going to have huge expectations beyond what we show you how to do. And generally speaking, I would say the vast majority of my students feel very settled and happy about the Hardy-Weinberg theorem when we're done talking about it. Okay, so let's just dive in. The Hardy-Weinberg theorem states that allele frequencies will remain stable in populations when the populations are large, there's random mating within the population, there's no migration into the population, there's no mutation, then there's no natural selection going on. So when you look at this, you actually realize that all of those assumptions essentially mean that there's no evolution going on within the population, right? That the population is just cruising along, the allele frequencies are stable, there's no deviation from anything here, no evolution's happening. And that's really important because in science, if we want to test whether a population is evolving, our hypothesis is not the population is evolving. Our hypothesis should actually be the population is not evolving. That's what we call a null hypothesis, right? And so the Hardy-Weinberg theorem actually helps us figure out whether the population is evolving so that we can reject our hypothesis that it is not evolving. Okay, hopefully that makes sense. Let's replay that. Think about it a little bit, but that's what we're doing. Okay, so the Hardy-Weinberg also provides a means of making predictions of what will happen if assumptions are violated. Okay, all right. So the Hardy-Weinberg theorem can really help us figure out, using an equation, what the allele frequencies are. Okay, and so we're going to call these allele frequencies P and Q of a gene. Okay, so we're going to let P be the dominant allele, big A, and Q be the recessive allele, so like a lowercase letter. Okay, so P plus Q should equal 1, if you think about it. Okay, so these should be the percentages of P plus the percentage of Q should always add up to 1. So if that's the case, here's the question. If P is 0.7, what is Q? Well, some pretty quick math. If you haven't figured it out yet, hit the pause button. If you have, we're just going to keep rolling. Should mean that Q is 0.3, right? Because 0.7 plus 0.3 equals 1. But this just tells us how many of each allele. What if we actually want to know about the genotypes of each individual in a population? Well, then we have to do just a little bit more complex math. Not too much harder, but a little bit. So what we're going to do here is we're going to get the individual genotypes. We know that we have 2 alleles for each gene, right? That's what makes up a genotype. So P squared is going to be the homozygous condition, or big A, and Q squared is going to be the homozygous condition for the recessive, so little a. So 2PQ is the heterozygous condition, which is equal to big A, little a. So if we take this equation, P squared plus 2PQ plus Q squared, it should equal up to 1, and that will give us each one of the genotypes. That's 100% of the alleles. So what you need to do now is either explain these two equations, how they're the same and different, to yourself, to your significant other, to a roommate, who's ever around you. Grab them and explain this. If there's no one around you, you can do it to yourself. And then also write these equations down. Okay? All right, hit the pause button, find somebody or yourself, and explain. Okay, let's keep going. And let's actually apply. So within a population of butterflies, the color brown, which is big B, is dominant over the color white, little b. And 40% of all butterflies are white. Given this simple information, which is something that is extremely likely to be on an exam, calculate the following. The frequency of homozygous dominant individuals and the percentage of butterflies in the population that are heterozygous. Now don't freak out. Some of you guys are sitting here going, "Oh my gosh, heterozygous, how am I going to do that?" Just hang in there. Okay, so here's number one. So we actually just need to calculate P and Q. We've done this already in our example, right? So 40% of white means they are homozygous recessive, right? Now this should be little b, little b. I apologize, it's little w, but just hang in there. So we would take the little b, or because of the typo, little w, and that's going to be q squared. So all we have to do is actually take q squared and work it out. So we'll do the square root of 40% equals a q of 0.632. So that just means if we go 0.632 times 0.632, we should get 0.4. You can do the math on your own and figure that out. It is correct. And since p plus q should equal 1, then p must be 1 minus 0.63 or 0.37. So 0.37, 0.632. You can check this. They should add up to 1, just to make sure you did the proper math. Okay, and they do. They add up to 1. Okay, so let's look at number 2, the percentage of butterflies in the population that are heterozygous. Now this is a little bit more complicated, but it's not too much harder. Okay, so we've got our P and our Q. We've already got these. So what is the percentage of butterflies in the population that are heterozygous? Well, this is really just 2 times P times Q, right? And that's going to add up to 0.47. So what is the frequency of homozygous dominant individuals? This is really just P squared. Okay, you have to calculate that. So 0.37 times 0.37 is going to be 0.14 or 14%. Your answer should add up to 1, so you can say 0.14 plus 0.47, right? So this is homozygous dominant, heterozygous, and homozygous recessive. It's going to equal 0.101, so pretty close to 1. So you know your answer is correct. Okay, hopefully that made sense. Hopefully that was pretty easy, and you can see this pretty clearly. Okay, so let's jump into genetic variation. So we all know that genetic variation, really, it's the raw material for evolution. And there are differences in DNA sequences. You get 2 major sources of variation in DNA sequence. You get mutation, which we've talked about, and you get recombination. Remember this from meiosis? And then evolution is going to act on these differences. But there's there's other major mechanisms of evolution that we need to talk about as well. So we've got evolution is actually what's going to be driving the allele frequencies, and in these 4 ways. So you have mutation, gene flow, genetic drift, and natural selection. And let's talk about each one of these. So mutations, we talked about these a little bit. Mutations are just random events. They ultimately, all variation comes from mutation. And mutations can be harmful, beneficial, or neutral, but the vast majority, just to remind you, right? They can be any one of these three things, but the vast majority are either harmful or neutral. Okay? All right, gene flow. So gene flow is really just the exchange of alleles between 2 different populations. Okay? It may result in new alleles being introduced. It doesn't have to, but it may. So here's an example here: brown beetles, green beetles. Brown beetle cruises over into here and passes those genetics around the green beetle population. We also have examples of human migration, passing alleles all around the world when we look at human migration patterns. Genetic drift is another one. So this is the result of a chance event. So just remember, genetic drift, chance event. It leads to random changes in allele frequencies, and we really mean this, random. Like these are things that you wouldn't expect. And generally, the smaller the population, the greater the effect of genetic drift. So what I want you to do is hit the pause button and just work through this figure to get by yourself or with whoever's around you, and see if you can make sense and see the genetic drift at work here. Okay, so let's come back together. So if you look at this, here's your population in generation 1. Okay, here's the allele frequencies. Well, if these are the only individuals here that produce offspring just by chance, then in your next generation, you're going to see that the allele frequencies have shifted dramatically. Okay, and if in this generation 2 only these individuals reproduce for whatever reason, then by the time you get to generation 3, you have no more heterozygotes, or you have only, yeah, this should not be heterozygote. That's a typo in the figure. But you should have this generation here with just the P, right? You should only have homozygous dominant individuals. All right. So here's some implications of genetic drift that we want to go over. So genetic drift is dependent on population size. And so here we have a small population and a large population. Just look at those two figures. Which one looks more stable? Which one looks kind of out of whack? Which one looks a little more crazy? Okay. So drift really has no direction. It's why it's called drift. Just imagine like a boat out on the ocean. It's just going to move wherever the wind pushes it. Okay. It's totally random. You're adrift. So drift can deviate above or below frequencies of the prior generation just on its own. There's nothing at work here other than just plain old drifting of genetics. Okay, and you can see that here, right? So when the population is small, you get these crazy whacked out where a gene can actually be fixated in the population really quickly when it goes up here to 100%, or that allele can be lost in the population here or somewhere in between, right? It could hang out for a while and then go to fixation. It could hang out for a little while and then disappear completely. Okay, it could also become close to gone in the population and then recover and go almost all the way to fixation, right? There's just a lot of drift going on. In larger populations, it's much more stable, but even then, over time, alleles are generally going to vary one way or the other, okay? Or at least they can vary one way or the other. All right, so here are some enablers of genetic drift. You have what's called the founder effect, and this really refers to an event when a small subpopulation migrates to an isolated region. Okay, so this is a portion of the population moving somewhere else. Just by chance, the genetics don't go, or they do, or they don't. Right? It just depends on who leaves and who's part of the founding population. And the bottleneck effect is a little bit different. So this refers to an event where a population is intact, but only a small portion of the individuals are able to pass their alleles to future generations. So here's your original population. You have this bottleneck effect where only these individuals are mating and passing on their alleles. So the surviving population is very different genetically from the original population. There's been no migration here. The population hasn't moved anywhere. The population is still in its original distribution, but for whatever reason, only a certain number of individuals were able to pass their genetics on, and you lose some genetics. Okay, so if we look at human migration patterns and movement, you can actually see that there's been founder effects multiple times in human populations, here, and then you can see the paths of migration, right, that have also happened around some of these founder effects. And then the source of the founder effect, right? So this is a source, okay, a source here, and a source here. All right. Here's a cool example of a bottleneck. Okay, so prairie chicken population in Illinois used to be up around 25,000 individuals. Okay, but over time, the grasslands were converted to other uses, and by 1993, less than 1% of the grassland that made up Illinois remained. Okay, and as you can imagine, there's going to be a decrease not only in the number of birds, but also in the genetic diversity. And here's a table that shows that. So here's Illinois. Here is before a bottleneck, right? So remember, this is a group of birds that had a large distribution, and without changing that distribution per se, there's no migration here at least. You had a lot of individuals that can only pass on. You had a subsample of individuals that can pass on their genetics. Okay, so this is pre-bottleneck here, 25,000. Post-bottleneck, 50 individuals. That's right, 50 individuals. Okay, so the number of alleles at any genetic loci, the number of alleles at 6 genetic loci, they had 31 in the original population. and in the bottleneck population, there's only 22. And you can imagine this is going to hurt the overall genetics in the group. There'll probably be a lot of inbreeding, things like that going on. So the percentage of eggs that hatch in the original population is 93%, and in this new genetically drifted population, there's 56%. And then when you look here, this bottleneck going on, there's no bottleneck that's happening in Kansas, Minnesota, or Nebraska, and you can compare the number of birds here and their hatch rates, and you can see that when the allele numbers are high, the hatch rates are high. So what we're seeing here is a bottleneck, a decrease in genetics, and thus an issue with the hatch rate. Okay, so a lot of you guys, you know what this is, right? Take a look at that. What is it? Well, it's the fastest land animal on Earth. That's a cheetah. Really amazing organisms. Genetically, however, they are incredibly similar, and that's across all of their distribution. So a lot of you probably think of cheetahs as just being in Africa, but you actually have some over into the Middle East and into India. These are their original distributions. These are their current distributions of where they're found today, so extremely retracted. And what we're actually seeing here is that what we're seeing among cheetahs is even among these different subspecies, the genetics are incredibly small. And this is part of the reason that it's incredibly hard to keep and preserve cheetahs, because there's not a lot of genetic variation. Now, this is probably one of the examples, actually, there's a lot of evidence coming in that this had nothing to do with humans per se. Now, certainly humans have had a huge impact on the distribution of cheetahs, but cheetahs themselves were already going through some sort of bottleneck, and the genetics were very low. So the question then becomes, do we try and pour a ton of resources into conserving the genetics of the cheetah when the cheetah was already most likely going to go extinct at some point in time due to a bottleneck that it had already suffered? Now, I'm not going to answer that. That's super complex. It's something for you to think about and try and decide on your own. All right, so how does evolution work? Mutations and recombination are going to occur randomly. Random events are going to sustain, support, or favor genetic variation. You have things like gene flow, genetic drift, and natural selection that are going to change allele frequencies. And then that is all wrapped up in meiosis and recombination. And this has what's really made all the diversity that we see on Earth today. And it's a fantastic process. It really helps explain a lot of the diversity that we see. And it's something that we can actually test and explore in science. It's really fascinating. Okay, and all of these things kind of come together, right? So we've got reproductive isolation, geographic isolation, polyploidy, hybridization, genetic drift, natural selection, recombination, mutation. All these things we've talked about are things that put pressure on this idea of speciation and can help to drive speciation. And that's where we'll stop today. Hopefully this lecture was good and informative, and I'm looking forward to doing the rest. We're in the home stretch now.